Welcome to Inert Chemistry. It is the third lecture of the topic DNA as a genetic material. In this last lecture, we will see about the Hershey and Chase experiment. This experiment provided a great evidence that suggested that DNA is a genetic material. In its experiment, they uses bacteriophage. Bacteriophage are viruses that infect the bacteria. The question is that why they uses these bacteriophages. It is the answer is they are having a very simple structure made of DNA and proteins only. So therefore, they uses these bacteriophages. These bacteriophages are also called T2 bacteriophages, and they infect E. coli bacteria that lives in intestine of mammals. In their experiment, they use radioactive isotopes of sulfur and phosphorus to tag the DNA and proteins. Here we see that how they do this experiment. In in their in their experimental setup, they first of all they take a solution. This solution containing radioactive sulfur. They allow the viruses to grow in this medium. You know that sulfur it is a part of proteins and not of DNA. So when they allow the viruses to grow in this medium, this radioactive sulfur incorporated into the proteins of the viruses. Then they infect the bacteria with these radioactive sulfur containing viruses. Then infect the bacteria. After that, they when these normal DNA containing and radioactive sulfur containing viruses infect these bacteria, then they uses a blender. By using the blender, they separate the empty protein shells of the viruses and only the phage DNA and bacterial cell is remain. Then they use centrifugation technique. When they centrifuged this material, the heavy bacterial cells they remain in the lower layer, also called pellet, and the lighter virus particles or viral particles they are coming in the upper liquid that is called supernatant. Then he uses or measured the radioactivity. They found that in when they uses sulfur, the radioactivity is shown in the upper layer. And you know that in the upper layer there are viral particles. In the next part of of their experiment, they uses you can say that a solution that containing phosphorus or radioactive phosphorus in it. They allow viruses to grow in this medium, and you know that phosphorus it is one of the basic component of your DNA that is involving in the formation of phosphodiester bonds. So as they grow the viruses in this medium, this phosphorus or radioactive phosphorus it gets incorporated into the bacterial DNA. These viruses then infect the bacteria. Again, they uses a blender and separate the empty protein shells and only bacterial cells and the viral components are remain. Then they uses the centrifugation technique. When they centrifuge this material, then again the heavy bacterial cells they form a pellet, and the lighter viral particles they come in the supernatant. And again they measure the radioactivity, and they found that in this case they found radioactive particles or radioactivity in the lower pellet and not in the upper supernatant. So from this they concluded that phage DNA entered the bacterial cell, but phage proteins did not. So, Hershey and Chase concluded that DNA, but not the protein, function as a genetic material of. Here is the slide added that helps you to know that how to write this as a long answer in exam. This is an assumption. Here again, you see that protein codes are radio-labeled first of all, but when the protein codes are radio-labeled, then no radioactivity is shown. But when the DNA was labeled, then radioactivity was shown. Thanks for watching this video. We will soon come with another topic.